So first we're going to have a presentation from Ben Rhodes and Snyder on restoring West Virginia's red spruce ecosystem. And after that, we'll have Dr. Steve McBride speak on volatile organic compounds, an overlooked carbon source for below ground microorganisms. So they're each going to present for about 15 minutes and then take questions, and then we'll turn it over to the other ones. And Breezy and Ben will be presenting together. So I would like to introduce both of them first, and then I'll introduce Dr. McBride for his presentation. So to introduce Snyder, for those of you who don't already know her, she graduated with a bachelor's degree from Ohio Wesleyan University in 2015, where she majored in pre-professional zoology. She spent the following 4.5 years in West Virginia working with the Nature Conservancy and partner organizations on red spruce restoration. This past field season, she worked with Dr. Barton and Green Forest's work on mined land restoration projects occurring in the Monongahela National Forest. She's now a first-year master's student in the Department of Forestry and Natural Resources with Dr. Barton as her advisor, and she'll be studying bat presence on the restored mined land in the Monongahela. So welcome, Breezy. And then Ben Rhodes. Ben graduated with distinction from UNC Chapel Hill in 2011, earning a BS in environmental science with a minor in biology. In 2013, he began his career with the Nature Conservancy in West Virginia, rising through the ranks from seasonal crew leader to coordinator of TNC's statewide ecological restoration program. And for his master's thesis project, he will return to West Virginia to study red spruce restoration work that he helped to facilitate. So without further ado, I want to turn it over to those two speakers for our first presentation today.
All right, thank you very much, Ellen. So I'm Ben Rhodes, and my co-presenter will be Breezy Snyder, and we are going to be talking about something very near and dear to both our hearts, which is the restoration of West Virginia's high elevation red spruce ecosystem. It's going to be a story of adaptive management in action. So just to show you where we're going today, I'm going to start out by giving you an overview of, well, what is the red spruce ecosystem in West Virginia? Then talk about the people who are working to restore it, the methods of restoration and the research that's gone into them. Then I'm going to get into this concept of adaptive management and, well, tell you what I'm talking about. And then I'm going to turn it over to Breezy to tell you, well, how we put adaptive management in action on the ground to restore red spruce. And then we'll wrap it all up with some results and conclusions. So the red spruce northern hardwood ecosystem in West Virginia. Red spruce is a keystone high elevation species, was once the dominant conifer species in West Virginia's high elevation forests, dominating over 500,000 acres and being an influential forest type on over 1 million more acres. This is a really unique, cool, wet, high elevation ecosystem that's really critical for wildlife. It's primary habitat for over 300 animal species, including rare, threatened, and endangered species, as well as secondary habitat for many hundreds more. And that's just on the local scale. West Virginia, of course, is situated right smack dab in the center of the Appalachian mountain chain. And so these high elevation, cool, wet forests are really critical for connectivity along the crest of the Appalachians, all up and down the East Coast. Unfortunately, as you can see from the map on the right there, these forests are in trouble. Where they once were present on well over 1 million acres, represented by the tan color in that map on the right, they've been reduced to fewer than 50,000 acres remaining, so between 5 and 10% of their original extent. And what we have left, as you can still see from the map, is very, very fragmented, just isolated pockets of residual spruce. And well, this story is going to be familiar to a lot of people on this call. Like so many of our other ecosystems, the industrial logging era of the late 1800s and early 1900s, it was a massive clear cut on a continental scale. It wiped out not just the red spruce forests, but also all the other forests from the central Appalachian Mountains. They were cut over, and then frequently they burned in massive wildfires. And well, what came back quite simply was not red spruce for the most part. That, of course, is a major ecological problem, both for the hundreds of animal species that depend on spruce and for that Appalachian corridor. And so there is a big network of people working to restore red spruce in West Virginia and the Central Appalachians. That partnership is called the Central Appalachian Spruce Restoration Initiative, or CASRI for short. CASRI is a network of over a dozen state, federal, and nonprofit partners, all working together on research, education and outreach, and adaptive management of the red spruce ecosystem. And of course, Breezy and I worked for one of those organizations, a nonprofit called the Nature Conservancy. Now, when it comes to adaptive management, there are three sort of basic techniques, and they're closely linked to what spruce is present on the parcel of land being managed. In the map on the right there, you can see sort of the spatial distribution of this. So in pink, those are our fragmented pockets of remaining high quality red spruce forests. And well, those areas are already okay. So they just need to be protected and in some cases improved, but generally just managed through protection and passive management. Just outside those, you can see these areas that have low spruce cover, and that's where we use silviculture to manage the forest for red spruce through a process called Spruce Release. Then outside of those low cover areas, those are the no cover areas that are suitable for red spruce, had red spruce in the past, but don't have it now. And in those areas, unsurprisingly, well, that's where we plant spruce. Now, Spruce Release, as of 10 years ago, was the big unknown. When CASRI got together and started thinking about how to manage these forests, we had a good idea of how to protect things. We had a pretty good idea of how to plant trees effectively. But these silvicultural techniques to release red spruce were pretty unknown. And so just the background of why this is necessary. So red spruce biologically is adapted to get established in the low light conditions of the forest understory and sit there and sit there and sit there for up to 100 years waiting for a canopy gap to form. And as soon as that light hits it, it shoots up, reaches for that canopy and succeeds into that gap. However, after the big cut of the industrial logging era, that ecological plan was not as effective. For one thing, without as many canopy spruce trees, there was a very limited seed source, so in many cases, it just couldn't get established in the understory. Then, even if it did get established, we have these much younger managed forests with much different gap dynamics to the old growth forests that red spruce evolved in. And so altered gap dynamics make it much more difficult for these spruce to get up to the canopy. So they just get stuck there for a very, very long time until they eventually die out. So human intervention is required, and that's this applied silvicultural technique, Spruce Release. There were decades of research that went into this. For starters, just after the big cuts, there were a lot of observation-based studies, especially in the Northeast, looking at regeneration of red spruce stands. Now, these had very little experimentation and manipulation. Again, basically just observation. But these researchers saw that, well, red spruce's natural recovery was very slow, and they really started to suss out these canopy gap dynamics. Then moving into the 2000s, as CASRI really started to think about how do we release these red spruce, we got some of our own researchers involved there in West Virginia. They expanded on those observation-based studies in the Northeast and started doing some manipulation, trying to explore how to restore that balance of red spruce and northern hardwoods by creating small selective canopy gaps. And well, that's what they went out and did. They made canopy gaps of various sizes and observed the results. And what they found was that they had really underestimated the size of the canopy gap required. They had wanted to make small, medium, and large gaps representing sort of the full spectrum of spruce response to that increased light. But what they found was that actually their largest gap was the smallest gap that a spruce will actually respond to. So they had not covered that full spectrum of the potential for creating gaps to release red spruce. And so that's where the adaptive management comes in. So all of that prior research, especially that CASRI project, gave us managers a really strong foundation for our work. That sort of set the baseline, that smallest gap size and the response we'd expect from that. And then, of course, general silvicultural practices gave us the supposed largest gap size that we could make. But then within that range, we didn't really know what to expect. And so we had these small specialized teams go out on the ground and actually do this work on a landscape scale, implement really rigorous monitoring protocols that then fed back into the implementation and that short-term feedback, and also started to suss out some of those knowledge gaps in the research and pointed out opportunities for further research. And so there is that self-sustaining cycle of research leading to implementation, leading to monitoring, which leads to more research, back to improved implementation, etc. And you can really see that in action with the work that we did. Now I'm going to turn it over to Breezy to talk about some of those monitoring projects.
Thanks, Ben. So we've implemented both some long-term and short-term monitoring. I'll talk about the long-term first, give a general descriptions, and then we'll get to the results after I discuss both of them. So for the long-term monitoring, the overall objective was to look at the long-term changes in forest composition and structure after a canopy gap creation. And this protocol was developed with input from Jim Wrench and other researchers. And so we looked on ArcMap about where this work, where we'd gone in and done this work, and we generated possible plots, using the ArcMap functions. And we then went in and only then chose the plots that actually had spruce release in them, with the obvious exception being the control plots, which we did not want that spruce release. So the selected plots were scheduled to be monitored 0 to 5 and 10 years post-treatment. And when we left TNC, we were actually in the middle of that monitoring protocol. So about only half of the plots had reached their 5-year mark. So we've been taking a lot of different data metrics, over 20, and some of that data we're looking at is basal area, ground cover area, dead versus live tree counts, spruce heights and diameters, and woody debris. So looking at the short-term monitoring, Ben created this along with some input from the CASRI members, and its intention was to be a quick turnaround in providing feedback to managers. It involves just 6 variables, 3 for treatment and 3 for the response. Because it was so simple, we could collect a lot of data and it could easily be analyzed. And it, answers the question, are spruce growing faster in response to the release? So that's kind of like the difference. Long-term looks at the overall forest structure. Short-term really just looks specifically at spruce growth. So I'll talk about the short-term results that we're seeing first. So on the graph that you see, the right 3 plots are the most important. The orange in the middle is the growth rates of fully released spruce. The gray in the middle is not released. And then the green over on the side is growth rates from natural gaps that we found. And as you can see, a fully released spruce has growth rates that averaged 235% greater than the unreleased spruce. And the growth rates of these fully released spruce are almost the same as the spruce that we're finding in the natural gaps, which is exactly what we're going for. We're trying to mimic that natural occurrence. For the long term, though, since we are still in the middle of our monitoring protocol, we don't have the full set of data. And so what we do have, it's quantitatively uncertain what we're looking at. However, we are seeing qualitative improvement of forest structure. There's a lot more coarse woody debris and snags without any massive devastation occurring to the forest. So I'm going to also talk a little bit about some of the other side projects, side monitoring projects that the crew was doing. So one of the advantages of being on or working with a field crew is their ability to be the eyes and ears of managers and researchers. One example of this was collecting some zombie moth samples that we randomly came across and sent to an interested researcher. And that's the picture you can see on the tree there. So these moths get infected with a cordyceps fungus, which attacks the moth's brain and forces them to land, and the fungus then sprouts out from the moth's body to spread its spores. And this kind of thing isn't something that you can pinpoint on a map about where you're going to find it. You kind of just have to wander around until you come across it. And we were already doing that wandering and could collect these samples for the researcher. And so another example, someone with a keen eye can see in this picture is also beech bark disease. So we, as I mentioned, roamed hundreds of acres. It took us far off the beaten path, countless hours. And as we, did our work, we would come across beech trees that have not been infected or are possibly immune to the beech bark disease. So we would take a GPS point and estimate a DBH. So when a researcher comes along that wants to get some, healthy beech DNA or possibly collect some beech nuts, they have a starting point using the database that we've already started. And the last thing I'll mention is at certain points during the year, we would help the Forest Service do their rare plant surveys. Some of our work would bring us, into these rare plant areas. And so we would help to make sure that what we were doing was not, harming these rare plants. So this is the kind of information sharing and gathering. It's one of the advantages to working in partnerships like Hazard and is part of helping with adaptive management strategies.
All right, and I believe it's back to me to wrap it up. So, with all of this put together, the research and the management and the monitoring that feeds back into management and research, that whole adaptive management cycle gave us this amazing project. Breezy already showed you the results. Release increases growth rate by more than double. And you can see here visually the effect that this spruce release has. So in those unreleased photos on the left, you can barely even see the spruce. They're these slightly darker green blobs, but they're in such deep shade that they're not going anywhere. They're going to sit in that shade and die without ever reaching maturity. You contrast that with the released images there, you can see we've really opened up that canopy, gotten a nice amount of sunlight down to those spruce. And as Breezy showed, that's really optimizing those growth rates, closely mimicking our goal of that natural gap creation. But even with all of that canopy removal and all of that light coming in, you can still see that all around those gaps, there are still those native hardwoods. There are even some remnant trees within the gaps. And so this is a very selective, very targeted process that maximizes our positive impact on the spruce while minimizing any non-target impacts for the rest of the ecosystem. And that would never have been possible without that adaptive management cycle of research to management to monitoring to research, etc. And that's our talk. So I believe, Ellen, that it is now question time.
Wonderful. Well, thank you so much. I really enjoyed that, hearing from you, Ben, and Breezy as well. And I guess it's kind of exciting because what you presented on today is prior to your starting as graduate students in our department, but a lot of that work is going to, or at least some of that work is going to be connected to your graduate research as well. Is that correct?
That's right. Not this work in particular. My graduate research is actually going to be connected to the planting monitoring, which was a very similar sort of adaptive management cycle. So it's the same general concept, still working with red spruce, but not on this release project. Still in the same ballpark.
And Breezy, you're going to be working with bats, right?
Yep. So still working on that, spruce landscape, but looking at the mineland restoration going on and seeing if bats are utilizing the landscape that we have, put back to rights.
Very cool. Do we have any questions? Feel free to unmute yourself and raise those yourself, or you can put them in the chat and I can ask them. I will say that I think this is illustrates part of what I was hoping that this session would do, and that sometimes when you go to a new place, your past research that is such an important part of your foundational kind of frame of things, isn't shared. And so I'm really happy that both of you could share that with us. We have a question from Dr. John Lodka. What was the size of the largest created gap in the study you mentioned? And Dr. Lodka, if you'd like to chime in and say anything else, feel free to.
I'm having trouble remembering the exact dimensions off the top of my head, but it was quite small. I believe it was something on the order of a 20-foot radius out from the spruce being released. So a circular gap and about a 20-foot radius. I can look that up and get back to you.
Any other questions? I enjoyed seeing the photo of the parasitized moth. I've run into some of those myself in the woods and always delight in them. So yeah.
Yeah, that was the biggest one that we found through our years of working. Yeah, they're always like a joy to come across, and we'd yell to each other in the forest, like, "Oh, we found one! You want to come and see it?"
Mm-hmm. Any other questions? Well, if you have other questions, feel free to put those in the chat or save them for the end and ask them afterwards, because both Breezy and Ben will stay on, but I don't want to hold off on introducing our next speaker. So join me in a big virtual round of applause for Breezy and Ben. Thank you.
Thanks.
If you have other questions, feel free to ask those. I know sometimes I'll be thinking about something for a little while before that perfectly formed question pops into my mind. So with that, I want to introduce our next speaker, Dr. Steve McBride. His BS is from Metropolitan State University, and his MS is from James Madison University. Then he did a PhD from Virginia Tech with Michael Strickland, and currently he's a postdoc with USDA NIFA fellow here with Rebecca McCulley in plant and soil sciences, studying how volatile organic compounds from cover crops affect soil organic matter formation in agricultural systems. And he's published in soil biology and biochemistry, ecology, biochemistry, and applied soil ecology. And I also have to brag on him in that he won first place award at the UK Society of Postdoctoral Scholars presentation contest, is that right? As well as first place for best visuals. So clearly a great presenter, and excited to have you join us today. So appreciate that, and nice to welcome you to our academic community here.
Thanks so much, Ellen. I'll go. I had never put that in my
I know it wasn't in my bio, but I knew it and had had to share it.
I mean, it's always a little scary, you know, when you set expectations high, because no presentation is going to be guaranteed to be good. But I really appreciate being invited to give this talk today. My background, I've primarily thought about ecology in forested ecosystems, even though now I'm currently working in like an agricultural setting. And so I'm going to talk to you today about volatile organic compounds and sort of what is their role in below ground ecosystems, because a lot of people haven't really thought about these compounds before. And so to kind of set the stage, well, before we do that, I want to thank everybody, because you get to the end of these talks sometimes and you running late and you don't ever want to forget to thank all your collaborators and undergraduate researchers that without them, several of the studies that I did during my PhD would not have been, you know, feasible. So, but I'm gonna go ahead and get us started with a little thought. Let's think about the last time you're in the forest, and in a department like this, I imagine that wasn't too awfully long ago. And maybe there was a recent rain and you're sitting there and you take in a deep breath and you smell that deep earthy smell of the soil. That's geosmin. It's a microbially produced volatile organic compound. And maybe you go on a hike through the forest and you end up at the top of a mountain and you're looking out at the Blue Ridge of Appalachia, and you gaze off into the beautiful cerulean haze blanketing those mountaintops. And what you're actually looking at is isoprene, which is a plant-produced gas or volatile organic compound. And so these volatile compounds are quite common in nature, yet they are not, their ecology isn't really well understood outside of, say, like plant insect signaling. And so let's start out with answering like a little basic question of what are VOCs. They're small carbon compounds, usually no more than 20 carbon atoms. They have low vapor pressure. So that means that they evaporate fairly rapidly. Think of like when you spray ethanol onto a lab bench, that's going to evaporate fairly quickly. That's a volatile compound. Now they enter the soil in multiple modes. So something like methanol could enter through root growth. So as roots grow, they release methanol. It's also the most common volatile compound to be produced during litter decomposition. Also, some roots will emit volatile compounds as some sort of like communication molecule. You can get atmospheric exchange, like that isoprene I was talking about, that will exchange with the soil and enter into the below ground soil matrix that way. And soil organisms like those actinomycetes will release compounds such as geosmin, which may be different sort of communication or defense mechanisms. But really, volatile compounds are a lot like dissolved organic carbon, something that we know quite a bit more about. So those compounds are also small. They don't necessarily have low vapor pressure, but they are often released into the soil through roots, through root exudation. They're not mutually exclusive, right? So some volatile compounds are highly dissolved in water. They're miscible, like methanol. It'll dissolve in water at pretty much any concentration. And then you can get different dissolved carbon compounds that it can enter the soil as they leach out of decomposing organic matter. And so these compounds, like I said, we do know quite a bit about DOC's ecology. And so one of the major mechanisms or like conceptual frameworks that kind of guide my thinking around this is the idea that external carbon sources fuel like below ground ecosystem processes. And so in this schematic here, We see that exudates and leachates enter into the soil DOC pool, where they are then accessible to the soil microbes. The microbes can consume them and build biomass, or they can respire off some of that carbon, or that DOC can be potentially stabilized directly into the soil matrix or indirectly through the microbial biomass. And we also know that these compounds have, pretty strong effects on ecosystem processes. Here is just a little graph illustrating that glucose addition increases enzyme activity, right? So this is the mechanism here, if you're curious, as you increase microbial biomass, more microbes in the soil means that they're producing more enzymes. And that's what we see here on the left-hand side. We have a couple of different treatments that are unimportant, but all of them for the control are lower than all the ones with the glucose addition. That it extends beyond simply affecting carbon cycling, though. So if we look at this this graph pulled from a study from last year from Ma et al., we see that when you add glucose to the soil, you actually decrease nitrification rates. So increasing amounts of glucose results in decreasing amounts of nitrification. And the thought process here is that as soil microbes have more available carbon, then they are going to have a higher nitrogen demand. And these heterotrophic microbes are better competitors for that nitrogen than the nitrifiers that are in the soil. And so that's what reduces the nitrification rates. And so we kind of come to this idea of like, well, could these volatile organic compounds, which are in many ways similar to DOC in how they enter the soil and like their lability or their like how simple they are, could they be functioning in the soil similar to DOC? And so above ground with you could have litter decomposition and those VOCs entering the soil that way, and below ground you could have other organic matter decomposition, or roots that are just alive and releasing carbon into the soil. And so we performed a stable isotope tracer study where we had 13C labeled leaf litter, and we published that this year in Ecology. And in this first panel here, I don't want you to focus on the different treatments, although if you're interested in getting more details, I have a QR code on the screen here that if you use your phone, it'll take you to the PDF and you can get the details on this specific study. But what I do want you to focus on is that up to 4% of biomass, somewhere between like 0.5% to 4% of microbial biomass was derived from volatile carbon that emitted from those decomposing leaves. And these leaves had no direct contact with the soil. So the only way for the carbon to enter the soil was through volatilization. And so this is, evidence that the soil microbes are actually consuming those volatile compounds. Now, in this next panel, we see that we also had enrichment in all of the treatments for the dissolved organic pool. And this could be, in one case, it could just be some of those volatiles that are able to dissolve in water. It also could be from microbial turnover. So after these microbes die, they release carbon into the soil through their cellular contents. And here we see that some of that carbon actually ends up getting stabilized in the soil matrix and becomes mineral associated. And then lastly, we see that the particulate organic matter also has a little bit of VOC derived carbon. And maybe this is coming from like cellular debris, cellular structures like, fungal mycelia and those sorts of things. And we also looked at how individual volatile compounds affect microbial activity and other ecosystem processes. And again, this is a different study we published in Biogeochemistry. If you're interested, I got a QR code for this one as well. And what you see in those in this panel, those two bars on the top are two different volatile compounds that are commonly released during litter decomposition, methanol and acetone. And you can see that there's a pretty striking difference in the respiration rates or the microbial activity like shortly after those compounds were added. And we also looked in this study, we looked at nitrate levels in the soil at the end of the experiment. And when I first saw this, I was kind of blown away, like how low the nitrate became in those two VOC treatments. So the addition of volatile carbon really drove down the available, or the nitrate in the soil. And we did some other work looking at the nitrifier community in this paper, and we saw that basically these compounds were inhibiting those organisms, either directly or indirectly, we're not sure. And so we've kind of established that these VOCs do act like DOC, and they fuel microbial activity and they affect nitrogen cycling. But it becomes a question of like, well, how important are they? Is there a situation where it makes sense to focus on volatiles? We know that roots exude a lot of DOC, and are these volatile compounds like important ecologically? And how do they compare like on like a rate level? And one place where this might be important comes, involves soil moisture. So in this image here from this Carminati and Vetterlein paper, I just want to illustrate how heterogeneous the distribution of water is in the soil matrix. And what we see is there are going to be places in the soil where microbes don't have access to water, and therefore DOC is going to be spatially limited. So either you're going to need to be close to the roots where you can get access like in the rhizosphere to carbon being exuded, or you need to wait for some sort of mass flow event like rain to leach carbon through the matrix to get access to it. And the reason that is that the diffusion rate of these dissolved compounds is quite slow. So about 600 micrometers squared per second. And so if you're outside of those areas, how do you actually get access to the carbon? Well, one option is volatiles. So the diffusion rate of methanol, for example, is about 15 million micrometers squared per second, which is 25,000 times faster than glucose. So a methanol molecule that gets released by a growing root can travel 25,000 times faster than a glucose molecule that is released through root exudation. All right, so to test this out, we did a lab experiment using, like an ecosystem in a tube approach. Had these falcon tubes with a small, cuvette inside the soil. And I added carbon weekly for 4 weeks. I had two different volatile compounds that I used. I used methanol and alpha-pinene. And just for the sake of connecting to the previous talk, alpha-pinene and limonene are volatile compounds released by spruce trees that give them their distinctive smell, or pine trees in general. Methanol, like I said before, is able to dissolve in water, while alpha-pinene is quite hydrophobic and does not dissolve in water. Glucose, I did two DOC compounds, glucose and oxalic acid. And these are just different, they kind of represent different classes of compounds released during root exudates. And I added them across a spectrum of moisture, so 25, 45, and 70% of field capacity. I measured respiration weekly. And at the end of the experiment, I harvested the soil, did some basic soil chemistry, and some microbial target gene sequencing. So let's jump right in. At the low moisture, the volatile additions actually increase microbial activity about 25 to 45% more than the dissolved carbon. So those are the two options on the right-hand side, the blue and the yellow. those are methanol and alpha pinene, they actually increase respiration above that of glucose and oxalic acid. When we go up to like an intermediate moisture, the alpha pinene respiration rate actually drops down while methanol stays higher. And at the highest moisture level, we see that essentially all of the respiration or microbial activity is about equivalent between all carbon additions. And so this is possible evidence that volatile carbon increases microbial activity more than dissolved carbon at low moisture, especially when you look at something like pinene, which is not able to dissolve in water and possibly is not able to diffuse through the matrix. And so when we looked at dissolved nitrogen, we saw that all like glucose, methanol, and alpha pinene are always reducing the available dissolved nitrogen. But at low moisture, we see it's about 20 to 35% lower in the volatile treatments. And at intermediate moisture, it didn't really differ between any of those treatments. And at high moisture, only the methanol and the glucose kind of pop out as being significantly different from the others. And so it's a little bit less clear here, but it does, C addition generally decreases dissolved nitrogen. And again, this might be due to that increased nitrogen demand by growing heterotrophic microbes. So we wanted to see, did this affect the community? And at low moisture, we see that methanol and alpha pinene both separate out from the control and the dissolved treatments, and the control and dissolved treatments don't really differ that much. The control and the two dissolved treatments don't differ that much. When we go up to intermediate moisture, that alpha pinene kind of moves back up and looks more like the dissolved treatments than it does the methanol. And at a high level, methanol is still different, and alpha pinene becomes even more like the control, while the glucose and oxalic acid are still a little bit different. And so it definitely is showing us that methanol changes the community a lot, while those those dissolved compounds maybe don't change it as much. And so here's everything together if you wanted to see what they all look like together. And we wanted to see, can we pick out individual taxa that could be driving these community changes or could be affected by carbon addition or responsive. So we did an analysis to see which carbon compounds or which microbial families were most affected by the addition of carbon compounds. And we picked out these 5 that accounted for over half of the relative abundance of the responsive taxa. And so in this next figure here, I'll take just a second to orient you. That line down the center of each of those treatments, across the top, we have our 4 carbon additions. The line down the center is like basically no different from the control. And then to the left of that line would be reduced below the control levels, and to the right of the line is increased above the control levels. And so there's a kind of a lot going on here, but the part that I really want to focus in on is that you can see that out of all of the different families that were responsive, a lot of them belong to this one individual phylum, the proteobacteria. That's not super surprising because we already know that proteobacteria oftentimes include organisms that are very responsive to low molecular weight carbon compounds. And then if we kind of like focus in a little bit and look across our moisture treatments, what we see here on the left-hand side, those first 5 boxes represent the low moisture, the middle 5 boxes represent the intermediate, and then the right 5 boxes represent the high moisture. And when we look at the methylophyllaceae, they are enriched in all of the treatments under methanol addition, but only under the low moisture conditions does alpha pinene enrich those organisms. And then the nitrospiraceae, which are involved in nitrification, we can see that they're actually reduced under carbon addition generally. And then these two at the bottom, the microorganisms and the pseudomonaceae, are responsive or increase in abundance over the control in both of the dissolved treatments. Although, as you can see from this, they increase at low moisture as well. And so I don't think we really see as big of a difference as we expected there. And then here, all of them, including the Burkholderia, and it kind of looks like there may be evidence of some C preference at different moistures, but really it comes down to, I think, that maybe DOC in this experimental setup, I wasn't able to isolate that effect as much, but potentially we're seeing that the volatiles are more responsive at that level, or at the low levels. All right, and so let's look at the next bit here, sorry. Kind of wrap that all up, we see that like these volatile compounds are definitely increasing microbial activity more than DOC at low moisture. Although unsurprisingly, all carbon compounds are increasing that microbial activity regardless of moisture level. We also find that dissolved nitrogen is decreasing from all of these treatments, but possibly more from the volatiles at, or at least statistically more from the volatiles at low moisture. And these VOCs and DOCs are affecting the community composition. Again, I think alpha pinene really jumps out as one that was most affected by moisture, where it's very different at low moisture and becomes more similar to the control as moisture increases. Much of the changes in the composition may be driven by these proteobacteria, like the Burkholderia, Methylophalaceae, and Pseudomonaceae, and these organisms are known to consume these low molecular weight carbon compounds. And so with that, I mean, together, I think that our results are starting to suggest that soil microbes may rely on different carbon types based on the local moisture environment. However, I think that our results support that they definitely are able to do that with volatiles at low moisture, although dissolved carbon maybe is more available to them than I thought it was. And so with that, I'm happy to take any questions, and thank you very much for your time.
Wonderful. Thank you so much for that presentation, and a lot of interesting research there. And I know that your research, this was mostly talking about your PhD research, but your postdoctoral work is continuing some of this, correct?
Yeah, so what I'm doing with my NIFA fellowship is I'm looking at different like the volatiles that are being released from different cover crops. Well, both the volatiles and dissolved carbon that leaches from decomposing cover crop material, and whether or not those compounds are increasing soil organic matter, and whether or not that's affecting like nitrogen cycling in agricultural soils.
Mm-hmm. Yeah, very interesting. I think both from that perspective, as well as thinking about our changing climate and how this might impact things there. A lot of implications of this work.
Yeah, definitely. And that's sort of some of what was driving the question around the moisture and whether or not, under dry conditions, are soil microbes going to have access to any carbon, or, is it going to be limited potentially to volatile compounds?
So do we have any questions for Dr. McBride? or our other two speakers? Okay, we have a question from Dr. Jen Yang. Great presentation. How much VOC would be released to atmosphere in our forests compared to VOC in the normal conditions, like the ones shown in your picture? How much VOC released to soil, and how would fire affect this?
Yeah, okay, so I can speak to some of that, and I don't want to quote specific numbers on too much of it because I don't have them at my fingertips. In a study out of the FEAR lab, Kelly Ramirez found that volatiles from decomposing litter can reach like the amount of carbon released in volatiles that are being emitted from decomposing litter can reach about 88% of the CO2 being released by litter, depending on the litter species. The amount of VOC that's released during decomposition is on the same order of magnitude of the amount of carbon that's released into the soil through root exudation, at least based on some like back of the napkin calculations. I didn't put that in here, but yeah, there's, you know, somewhere in the same order of magnitude for VOC released from decomposing litter versus root exudates released in the soil. Now, how much of it makes it to the atmosphere is really, you know, dependent upon a lot of local, you know, like microclimatic conditions. So humidity, like local humidity affects VOC release as well as temperature and moisture. So, and fire, I can't speak too much of it. I mean, obviously there are going to be some issues with like burning that litter layer up, and that's going to probably change a lot of those microclimatic conditions. And I imagine that affects the amount of VOC that gets released. I hope that answered your question. I'm sorry if I didn't completely answer it.
Great, and feel free to unmute yourself if you have further questions. I want to raise this one from Dr. Chris Barton. What implications could this have on bioremediation of VOC contaminated soil? Think leaky underground storage tanks. Keep the soil dry to maximize treatment, for example?
Yeah, so this is like an area of VOC study that I haven't really put a lot of thought into, because and it's kind of weird, I guess, because that's where most of the research is in this is VOCs that are toxins and VOCs that come from like petroleum products and that sort of thing. And I think I don't want to say anything about it that could be really, really wrong. But I would imagine that, yeah, dry soil would allow them to volatilize more because they do evaporate when the water level is low. And so you might get more soil, or you might get more of it to volatilize out of the soil and just, you know, enter into the atmosphere. I don't know if that accomplishes the goal or not, but I would imagine it would at least get it out of the soil if that is the goal.
Well, Chris, if you got any further questions on that, feel free to unmute yourself. Anyone else have other questions, comments, things they'd like to follow up on?
Yeah. Hi, I study wildlife, so I'm well out of my field here, and I have a pretty basic question. Why do we care about microbial activity? How does that fit into the larger ecosystem?
Yeah, well, sorry for not making that clear. So I use microbial activity as a kind of a way to simply understand the idea of just soil respiration. And so microbial activity, in this case, we're talking about specifically just the amount of CO2 that's being released to the atmosphere, right? So we're going to be, I mean, that in and of itself is important from a carbon cycle standpoint. But there are some other things that we can kind of infer from that as well. And I hinted at it early on with the glucose additions, is that when you increase microbial activity, that oftentimes is concomitant with an increase in like microbial biomass a lot of times, which means that that ecosystem process rates are changing. And so if you're producing, say, more extracellular enzymes, then you're decomposing organic matter more rapidly. And also, from a nitrogen cycling standpoint, when your heterotrophic microbes are more active, they're competing for available nitrogen with the organisms that, you know, nitrify and do other nitrogen transformations. And so, I mean, that's the 30-second answer. Does it answer your question? I mean, I may not have made you care, but I hopefully answer the question of why it is important in some ways.
Yeah, you at least gave me some terms to Google to like learn more. So, yeah.
Excellent. Well, I want to thank all of our speakers today. Steve, Ben, Breezy, really appreciate you taking the time to speak with us about your work. Some great presentations. So join me in giving them a big virtual round of applause. Not the same as in person, but still pretty good. Lots of hands clapping. I know that we will be off for seminar next Wednesday. But the following Monday, November 30th, we have our final seminar of the semester. It's another joint seminar with UK's Department of Plant Pathology, and it's during their normal seminar time. So if you're wondering about that weird time, that's what that is. November 30th at 3 p.m. And it's with Monique Sakalidis. She is at Michigan State University and focuses on the epidemiology and biology of oak wilt, as well as other plant pathogens, but she'll be focusing on oak wilt for her presentation. So it should be a great talk and something that I know a lot of us are interested in. So hope to see you then. And thanks again to our presenters, and thanks to all of you for joining today.